Chapter 3, Section 14, Amino Acids and Nucleotides. As I said in the previous section, you don't absorb the nucleotides, the direct genetic material of whatever you consume. No, you synthesize your own. You synthesize your own amino acids, unless it happens to be one of those we refer to as the essential amino acids. You know, the essential amino acids, the essential vitamins, are the things that we humans, or we some mammals, we some animals, just do not have the genetics, the blueprint, the instructions on how to make. Thus, we have to consume them. When it comes right down to it, we have biosynthetic pathways to make most of the amino acids in us. So those that we do not obtain from our environment, we will synthesize from glucose or other carbon sources. Okay, if you can make one of the amino acids in a certain family, because remember, some of them kind of look the same, leucine, isoleucine, things like that. Well, they're going to have shared steps. You make one, then that product gets splits off and now can make others also, based on that one as a precursor. Got to remember, when it comes right down to it, this is all carbon skeletons. You know, an amino group, an alpha carbon, a carboxyl, and then some form of an R group off of the alpha carbon. So glycolysis, you know, I've said in a previous section, nothing happens in a vacuum. So you look at glycolysis, you take the pyruvate looks like the alanine family. Valine, alanine, valine, leucine. You take the 3-phosphoglycerate, looks like the serine family, and glycine and cysteine. Phosphoenolpyruvate plus some other things from the pentose phosphate pathway. This is one of the intermediates. We didn't go over all the steps in the last section. Gives you what is referred to as charismate. Charismate is part of the aromatic family, those that have an aromatic side group. Phenylalanine, tyrosine, tryptophan. You take the alpha-ketoglutarate and oxaloacetate from the citric acid cycle. Here's the glutamate family, chlorine, glutamine, arginine. The oxaloacetate, boom, 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 boom. So like I said, all you really need is something with an amino group, NH2. Originally, this is going to come from inorganic nitrogen, ammonia, nitrate, that one organism is able to fix and then have it enter into their metabolism. Glutamine dehydrogenase or glutamine synthetase will take ammonia and then start the formation of glutamate or glutamine. Glutamate, we do not have this ability. Glutamine dehydrogenase, glutamine synthetase. We don't have this. This is going to be plant. This is why some of these we have to eat, the essential amino acids. Once you take up and you start breaking down the proteins to polypeptides, polypeptides to amino acids down to their components, you can strip off that amino group and then add it to whatever you need to in your normal, anabolic metabolic processes using a transaminase or an aminotransferase. Alpha-ketoglutarate plus an ammonia gives you glutamate. Glutamate plus another gives you glutamine. Glutamate plus oxaloacetate, alpha-ketoglutarate, and aspartate. Starting to see the pattern here. These ammonias, if it's a microorganism, they take gaseous nitrogen, like we've talked about in a previous section. For those of us who can't fix nitrogen, this comes from the diet or things that have been absorbed by that cell. That compound gets consumed, and then you have some form of a transferase that will then transfer it to your target compounds. The making of our purine and pyrimidine nitrogenous rings is a very complex process. And no, we're not going to go into all the different steps because this isn't cell biology. This isn't biochemistry. Its carbon and nitrogen sources are going to be cut, pasted together to form these rings. You know, the nitrogens are going to have different sources. The amino group of aspartate, the formal group of folic acid, the nitrogen from glycine, carbon dioxide, nitrogens from glutamine. There you go. You're going to start off here. Aspartic acid, carbon dioxide, ammonia forms ordic acid, which will then be modified to form uridate, basically uracil, which is the precursor for the making uracil, the making of